Okay, ready? Okay. Hey guys, welcome to Mechanisms of Cancer. We are just reviewing your Pingry experiment, and we thought we'd give you some feedback. Since we had a lot of it, we thought we'd give it to you in video form. So here we go, Dr. Carr.
So the first thing I want to address is general writing problems. First of all, concision. Write only what's being asked. A lot of you provide a lot of superfluous information. I think this can get you off track and it confuses the reader. You also, a common problem is repetitive wording. A lot of times there'll be 3 sentences in a row that are all saying basically the same thing. Just a general writing critique, especially in science, is you want to aim to be concise. Clarity is also a big problem. A lot of you had very awkward sentence structure, which can detract from the meaning, and kind of strange vocabulary choice at times. A general tip is that you want to proofread and that it's good to read things out loud. That this way you can tell when you're reading out loud if a sentence runs on or if you're missing a verb, for example, or if it's a fragment. Along the lines with the vocabulary problems is a problem with imprecision in your language. You want to minimize your use of pronouns. The word it appears a lot, and most of the time it's unclear as to what it is referring to. And if you choose a vocabulary word, specifically a science word to use, make sure you know what it means and that you're using it precisely and properly. A lot of times it's unclear if you really don't know the difference between, say, a gene and an mRNA or a protein, or if you're just being sloppy with your language, and it's hard for us to try and determine that difference. So be careful with loose terminology. As far as factual information in the first question, the biggest problems were understanding mutations and what they can do or don't do, and exactly what RNAi is influencing in the cell.
So yeah.
That's question 1.
Okay, so I think just to review there, I think one of the 2 most important things that Dr. Kirkhoff mentioned was making sure that you proofread your answers and making sure that you read them out loud. That will clear up a lot of the problems. So I graded the experiment, excuse me, I graded the experiment, the second part of the question, and one of the biggest problems I found was causalities. So a lot of you said that if you find a mutation in the tumor gene, or the Pingry gene in the tumor, that would cause the tumor. Well, you don't know that. Just because you find a difference in 2 things doesn't mean that that difference is the cause for the difference, or for the tumor, for example. So one thing that you can do is you can kind of think forward. So if there was a tumor and you suspect that a specific mutation is the cause of that tumor, what you can do is induce that mutation in a future experiment, or in if you have 2 clones, 2 mice that are the same genetically, and you give one that same mutation, and that mouse then generates that tumor, what you now have is much stronger case for the fact that the mutation causes the tumor.
And in problem 1, a lot of you wrote about the causality of the missing protein, which, as we just mentioned, hasn't been shown. There's no evidence for that, but it also was not necessary to answer question 1. So again, with the superfluous information.
So an example of exactly what a wrong answer would, I have a cancer tumor and I have regular tissue. I take samples of both of them. If I find a mutation in the cancer tissue, then I'll know that the mutation caused that tumor. That's just not great logic. You guys used a lot of techniques in your experiments, and that's good. The problem is that you used a lot of techniques wrong. Anytime you look up a technique, you want to make sure that you find a source for it. So you want to find somebody that's used that technique before, whether it be sequencing, microarrays, southern blotting, northern blotting. You guys use several different techniques. You just want to make sure that I know where you're getting that technique from, and in case you use it slightly wrong, I can actually go back to the place that you found your information from and find out how they used it. And in some cases, I might not be familiar with the technique that you've mentioned, and I want to make sure that I'm not marking something wrong just because I don't know about it. A few other things, techniques versus experiments. So a technique is a procedure that you will do to basically help you answer an experimental question. There's a big difference between the two. And I think a lot of you guys focused on techniques more than experimentations. So you focus more on making sure that I understood that you knew what sequencing was, but then you didn't use sequencing properly to kind of answer the question, does this mutation, for example, cause the tumor? So we didn't want you to explain the technique in the second part at least. We just basically wanted you to use that technique and help us to answer the experimental question.
And first you have to define your experimental question.
Good point. Let's see. We've got no its. A lot of people assumed that if there was a mutation, there would be no gene present. And you have to think mechanistically in an experiment, which means you have to think about why a mutation would cause no gene. So for example, if I have a gene and I change one nucleotide in that gene, it doesn't necessarily mean that the resulting protein will not appear. It could be that the resulting protein appears with an amino acid change. It could be that there's absolutely no change in the resulting protein. So just saying, for example, that there's a mutation isn't sufficient. You need to say a mutation in this part of the gene, for example, maybe a stop codon or a start codon, or in a promoter region, might cause a significantly different formed protein. I think what we're seeing is that you're not quite thinking the entire thing through. We need you to be precise. We need you to go all the way through gene expression every time. So we've harped on this idea that you have DNA to RNA to protein, and we need to see that in almost everything that you write. So if you are saying that there's a mistake in a gene or in the DNA, and then immediately just assuming that there's going to be a result in the protein, that's not sufficient. You need to get us from the DNA through transcription, through translation, and if there's splicing involved or whatever, you need to get us from that beginning point to the end. Again, just be more precise. I think one thing that you can do to really help would be to chart out your answer before you do it, especially with the experiment. Say, you know, I suspect this is the cause, this is the effect of that effect, and all the way down to the lack of protein. Diagramming will help you a lot. I know in the future when we grade these, we're going to be a little more strict. I think we were using this more as a learning exercise than anything else, and so we were being very forgiving in our grading. So make sure that you take this information and that you apply it to your future writing.
Sounds good.
All right, great. Bye. Okay, you want to do it again?
Sure.
Yeah.
I think it's fine. I stumble on my words a few times, but
I think it's good.
I think it's fine.
Okay, let's do it.
It serves its purpose.
Yes. And
And I've classed
served it quickly. But it's amazing, you know, it would have taken us an hour.
Mm-hmm. No, this is good.
Write that. Uh-oh.